I'm going to switch gears to more the theme of endogenous regenerative medicine from endogenous cells. And I'd just like to acknowledge my co-authors. A lot of this work was generated by my postdoc, Shan Lee. From Montreal, you can see we have our campus on the top of the hill. Bone, it's a porous living material. This shows a micro-CT of subchondral bone, where you can see the porous architecture and mature bone that has the bone marrow and endosteum, which is the here are these cells that line the inner surfaces of the bone. And when bone is wounded, we know there are mesenchymal stem cells in the bone marrow that become activated. And here you can see the formation of new woven bone. There are two regenerative responses in damaged bone. One is to generate, with osteogenic induction, the formation of osteoblasts on the woven bone surface. They can build up off of a pre-existing bone. Or here what you see is regeneration of bone through cartilage formation, and in certain contexts the bone will receive a chondroinduction signal, which allows the development of cartilage from the surface of the bone. And in this bottom figure, what I'm showing you is a healing osteochondral defect, where the bone has been stained with von Casa, which stains the mineral, the phosphate mineral, and here the toluidine blue is staining metachromatic glycosaminoglycan in the developing callus. So when bone is wounded, it will spontaneously regenerate through mesenchymal stem cells that differentiate to osteoblasts or chondrocytes. And the end stage, this cartilage will calcify and become bone. Actually, in some cases, the bone void is too big. This is what you call a critical size defect, and the tissue that repairs in the defect is neither woven bone or callus, giving rise to calcified tissue. It can become a fibrous repair, and in fact, the part of the motivation I had for doing this project was someone I met at a Canadian Arthritis Centers of Excellence annual meeting. Her name is Francine, and she had had this pressure building up in her chin, and she didn't know what it was, and it turns out it was a tumor, and so they had to take out her mandible. It was an advanced osteosarcoma, and to repair that, they took her fibula. Actually, the most common repair for these things is they take your own bone and transplant it into your jaw. And luckily, the graft took on one side, and she had a replaced mandible with her own bone. But on the other side, she had a non-union. This is a non-union. And they fixed it with metal plates. And at the time of the meeting, she explained to me that the metal plate was cracked. The bone was not healing, and she was pretty scared. She said, I don't know what's going to happen. And what we decided is, with the colleagues, is that really this kind of non-union could benefit from a kind of tissue engineering approach to help regenerate the bone. And the principal aim of bone tissue engineering is where you start with a wound where you're going to have some bleeding, and hopefully what you can do is replace that with a porous scaffold. And in the idea behind endogenous repair is that this endogenous scaffold is going to allow the ingrowth of bone. So you're going to get ingrowth of hopefully woven bone with, of course this won't take place without invasion of blood vasculature, but eventually the porous material would be allowed to degrade. This is of course a cartoon, it's not what really happens in real life yet, but the bone is overtaking the porous implant that's degrading slowly over time along with vascular invasion, and eventually would be replaced by living bone. So this is the real goal of bone tissue engineering, is to reestablish living bone. So one approach that actually a lot of people have been studying is the use of a bioplastic called polycaprolactone. It's a polymer created through that can have a variable chain length. In fact, it only melts under high temperature. You can use an approach with a porogen polyethylene oxide, which is biocompatible. And in this process, what we showed is that if you melt these two polymers together and then do this static annealing, if you let it anneal for a longer and longer period of time, you develop a pore structure that's controlled and the pores get bigger and bigger. So we could make porous scaffolds for these bone tissue engineering applications. And the next stage of the study was to proof of concept that these scaffolds could support bone differentiation. And to do this, we use an in vitro osteogenesis assay, and actually in 3 dimensions. But the normal assay is you seed mesenchymal cells, primary human mesenchymal stem cells from bone marrow on the surface, and after 10 days, they'll become confluent. And then you put the osteogenic medium, which contains dexamethasone, on the cells and let them grow for 3 weeks. You have to let them grow for 3 weeks because it takes about a week to start laying down collagen, and then another 2 weeks for the collagen to start mineralizing with the mineralizing enzymes and factors in the matrix. But what we found was, in fact, the PCL surfaces are quite hydrophobic, like any plastic. The cells don't spread or adhere to the plastic. And when we cultured in 3 dimensions, what we saw was that the cells will grow into the scaffold. Here, the H&E stain, you can see the pink stained cells. But there were very few cells actually growing on the surface of the scaffold, here in S. They were mostly growing in the pore spaces. And when we did an alizarin red stain, we find that the mineral was only accumulating in areas where there were pockets of cells. So in fact, these scaffolds are not osteoconductive. They're not allowing bone growth on the surface. So there's a major challenge in designing surfaces of these bioplastic materials for bone tissue engineering. We need an osteoconductive surface. That means that the surface allows cell adhesion, osteoblast differentiation, which leads to mineral deposition, and ideally, in the case of dental bone voids, the surface should resist infection as well. In fact, surfaces guide stem cell responses, and actually you can't really see very well here. This is the vinculin stain showing focal adhesions that are formed on a fibronectin-coated plastic compared to just tissue culture plastic. So depending on the cell surface, the stem cells will spread and develop focal adhesions or spread and have different cell responses. So in this project, actually was carried out in a collaboration with a small biotech company in Winnipeg called Kane Biotech. And they developed a concept that you could fuse P15, which is a biomimetic peptide. Maybe you've heard of it. It's a 15 amino acid peptide that's derived from collagen type 1, and it can stimulate osteogenesis in vitro by mesenchymal stromal cells. And CSP is an amphiphilic peptide that actually is expressed by bacteria. And it's a quorum-sensing peptide that has an isoside that's positively charged and hydrophobic. And this synthetic peptide actually has membrane perturbation properties. It can perturb the membrane and kill bacteria, and it can also inhibit biofilm formation in a mutant form. So the structure of there's an alpha helix that has a cationic side and a hydrophobic side. So the hypothesis we tested in this study was that P15CSP, it's a novel fusion mimetic peptide, could have emerging properties that aren't shared by CSP or P15 alone when it's deposited as a coating, so the fusion peptide. In this study, we used a dry coating approach, which actually is more translatable to a medical device. because once you've dried coating, you can store it. It's stable at room temperature. So we simply filter sterilize the peptides and let them evaporate in the cell culture hood overnight. And this was allowing easier collaboration because we could simply ship the plates to Kane Biotech to perform the biofilm assays. And in this bacterial culture and biofilm assay, they inoculate with Streptococcus mutans, and after 18 hours of culture, you can measure planktonic growth, that's the liquid phase growth, or biofilm formation through a dye stain. And then we use the same types of plates to look at osteogenesis. We analyzed adhesion, cell spreading metabolism, and then we looked at osteogenesis using the traditional assay. So these are the results of the antimicrobial assay. And you can see by the bar in gray that the CSP showed a killing effect on the bacteria after 18 hours. That when we deposited mixtures of P15 and free CSP was more enhanced. The P15 CSP fusion peptide coating showed a decrease in biofilm formation, which you can see here, less biofilm on the surface of the well. And the free CSP was able to inhibit biofilm formation at levels that also inhibited planktonic growth. So this part of the study showed that the coating in fact inhibits biofilm formation without inhibiting planktonic growth. The free CSP coatings inhibited planktonic growth in higher concentrations, also inhibited biofilm formation. But the P15 peptide by itself had no effect. In our cell adhesion assay, we exposed the mesenchymal stem cells to the coated Petri dishes, and after 1 hour, the cells were washed off, and this shows a phase contrast where the cells stuck very well to the coated surfaces with CSP, P15, and fibronectin. There was an increased adhesion with P15, and after 1 hour, we see the cells have started to spread on the CSP, P15, CSP, and fibronectin surfaces, and not so much on the P15 and uncoated surfaces. After 24 hours, we see good spreading for these surfaces and a little bit less for the P15 and tissue culture plastic. When we performed a MTT assay, we see the uncoated P15 and P15 CSP are all cytocompatible, whereas CSP shows a slight cytotoxicity for mesenchymal stem cells. So this part of the study showed that the fusion peptide, CSP, and fibronectin accelerate surface attachment in serum-free media. And these coatings are cytocompatible and promote rapid restructuring of actin and spreading. In our osteogenesis assay, I don't know if you're familiar with the osteogenesis assay, but typically we use 2 markers to show differentiation of cells to osteoblasts. So the alkaline phosphatase is an enzyme that's used by the cells to generate biomineral. It degrades beta-glycerol phosphate to phosphate and glycerol. And alizarin red is a stain for calcium. And here you can see the deposition of a mineralized matrix when the cells are cultured on the P15 CSP coating, along with alkaline phosphatase stain in all of the wells. And you can actually elute the alizarin red and quantify it by absorbance. And this assay showed that the coatings with P15 and P15 CSP were able to increase calcification of the matrix in the presence of dexamethasone, and there was a higher average calcification with the fusion peptide. The next stage of the study, we were interested to see whether or not we could obtain the same effect of the coating on thin films deposited by plasma-enhanced chemical vapor deposition. It's also called PECVD. And the way this works is that you take a ethylene gas mixture and argon oxygen, and under a plasma polymerization, you can deposit 200 nanometer thick films, and we showed that these have 6.7% carboxylic acid content. We also generated thin films that were hydrophobic from hexamethyl disiloxane vapors, which were generated on tissue culture plastic. And the idea behind this was that the anionic surfaces, these anionic hydrophilic surfaces, could better retain the CSP peptide, the fusion peptide, through the CSP cationic isoside. Actually, at the time of the experiment, we thought that the hydrophobic surfaces may retain the peptide as well through the hydrophobic patch. But as I'm going to show you, this wasn't going to happen. So we characterized these surfaces through water contact angle. You may know what this is. It's when you put a drop of water on the surface, and if it's hydrophobic, like Teflon, the water contact angle is greater than 90 degrees. And if the surface is highly wettable, the water contact angle is less than 90 degrees. So actually untreated is tissue culture plastic. And your tissue culture plastic normally has been treated by normal plasma under CO2, which gives an anionic surface. And this has a water contact angle less than 90 degrees. The HMDSO has above 90 degrees, and the LPPEO surface is even more wettable. And when we culture MSCs on these surfaces, we find a very spindle-like morphology on the hydrophobic surface and a more spread morphology on the LPPEO surface. And I was just interested this morning at the, who's the speaker? Affymatrix, Shirley, yes, Dr. Shirley's talk, that perhaps the cells could be undergoing asymmetric division on this type of surface. So this is where we had a quite interesting finding. On the osteogenesis assay carried out on the hydrophobic surfaces, the cells actually were able to adhere and proliferate and to express alkaline phosphatase. But after 3 weeks in osteogenic media, the cells have not produced any calcified matrix. And when we carried out the same assay on the LPPEO surface with the different peptide coatings, what we find is the cells have all expressed alkaline phosphatase and heavy calcification with LPPEO. And the quantitative assay, we see an enhanced calcification compared to the tissue culture plastic coated with peptide, and the best calcification was with the fusion peptide. So these results showed that all surfaces, with or without the peptide coatings, permitted the mesenchymal stem cells to differentiate to early osteoblasts by alkaline phosphatase activity, and the hydrophobic thin film suppressed calcification, the biomineralization, with or without peptide coating, and that these P15 and CSP and P15 coatings on hydrophilic surfaces were able to enhance the biomineralization in osteogenic media. The study was limited by, in fact, we have only indirect evidence that this coating was stable, but we did have a reproducible cell response, which leads us to believe that the coating, we did demonstrate bioactivity of the coating, but there is a possibility some of the peptide solubilized during the assays and led to a combined response to soluble and immobilized peptide. So the conclusions of this study are that the hydrophobic surfaces, in fact PCL, like PCL, prevent MSCs from biomineralizing, and it may be because they cannot reach a highly packed density that you need for osteoblast function. The fusion peptide has properties not shared by P15 and not shared by CSP, so this fusion peptide does have emerging properties, and it may enhance osteogenesis by promoting cell adhesion or by promoting cell proliferation on hydrophilic surfaces. And I'd just like to acknowledge my collaborators at Kane Biotech, Mike Wertheimer, who does the plasma modification of the surfaces, Greg DiCrescenzo, who helped co-supervise Shan Li. Thank you. Thank you.
Is any question? One over there.
Sir.
Please.
Yeah, next. Research. So they can
I'm just wondering. Yeah, I'm just wondering. Uh,
Anyway, as we work. Go ahead.
thank you. You know, the fibronectin, right, is a adipokine. Do you have a think about what the mechanism underlying the fibronectin enhanced the mesenchymal stem cell attachment to the film?
Well, it's integrin receptor mediated.
That's when, you know, the adipokine, normally mesenchymal cell can differentiate to adipokine. No, they release a lot of adipocytes, but release a lot of fibronectin. This is the reason I ask you, because we are run through the mesenchymal stem cell genesis. We found a lot of fibronectin. Actually, negative feedback inhibit the attachment to the plates, I would say. So this is a mechanism we still don't understand yet. So I just wonder whether you have any thoughts of it.
Unfortunately, we didn't have fibronectin in our osteogenesis assay, so we didn't study that. Yeah.
Thank you very much. Okay, there is, so let's go.